forward. And why don't we just finish up the lecture from Wednesday and then hop into lab, okay? So let's see, where am I? I got my slide, yeah. Here we go. What's going on? Okay. So we left off with staining, basically. We said that there are different types of stains that give us different types of information, and some work better than others depending on the application. If you want to look at live specimens or fixed specimens, then these stains will be better or worse depending. We said that there are basic dyes that are positive stains, and so they specifically stain the microbe. We then said that there were acidic negative stains, which hopefully this image here looks familiar because you just recently saw it. That would be a negative stain. So it stains the background, but the microbes repel that chromophore or a chromogen. And so it's white on a dark background. We said there were simple stains that just use one dye. There are differential stains that help us to distinguish between bacterial species based on whether or not they uptake those stains or not. And we said there are structural stains that stain different parts of the bacterium to show us whether or not it has things like flagella or a capsule. Here are some examples of a simple stain. On the left is a methylene blue stain at staining the microbes. On the right is a negative stain, so it's staining the background, not the microbe. And we talked about differential stains. Gram-positive is by far the most common stain. And there will be a Labster simulation, I believe, where you do a gram stain and shows you how to do that. There's the acid-fast stain. Again, if it stains at one color or another, that tells us about the biology of the bacteria. Spore staining shows us what stage it's at sexually, whether it's a spore or what's a vegetable cell. Capsule stain, that's going to be the structural stain, right? It repels, the capsule repels the dye, and so you can see the halo. Flagellar stain shows us these flagellar structures, and it's putting the cart before the horse a little bit because we haven't talked about morphology yet of bacterial cells, but we'll get into that later on. Fluorescent stains, we talked about, they stain specific parts of the cell, and so you can really home in on regions of interest based on using different stains or different antibodies that are conjugated with a fluorescent tag in order to see those structures in the cell. And of course, we talked about the 6 I's: inoculation, isolation, incubation, inspection, information gathering, and identification. So for the homework, if you guys haven't already gotten to that, the last question, I believe, is asking you to use the 6 I's to describe how you would culture something and identify it from a urine specimen. And so I just wanted to go through the 6 I's one more time just to get it firm in your heads and, you know, show you with pictures and images kind of what the process is. So you have isolation, you have individual bacterial cells, and this is, again, so we've already inoculated and we're going into the isolation step where we have parent cells and we have 2 different types. In this case, we have the yellow circular ones are going to be a cocci bacteria, and then these red rod-shaped ones are going to be bacillus bacteria. And so they go on the plates and they then grow up and form these colonies. And those colonies, then we can pick out, and that's going to separate it. Either if we want the red or the yellow, it's going to separate out. Another way is to get those colonies to separate out is to streak the plate. And unfortunately, we won't be streaking any plates. I guess you'll be inoculating a plate for one more of the take-home labs, but you won't be isolating anything, unfortunately. But there's a method to it. What you do is, let me get my annotation here. You are going to, usually you get a pipette and you pipette a little bit of the sample onto one corner of the plate right here, okay? And so that pipette has a liquid culture of your bacteria. And so then you take a loop, which is a metal wire. It has a little loop at the end, and you'll flame it under the Bunsen burner because you want to kill off any microbes that are already existing on that loop, because, right, microbes are ubiquitous. They're everywhere. And then you'll use that loop to just barely touch some of the inoculum that you put on that plate. And then you'll streak it side to side like this. And then you flame it again to kind of kill off, in this point, you're not killing off contaminated stuff, but you just want to, the whole purpose of doing this is to dilute the bacterial cells. So in culture, it's going to be very concentrated. And what we're doing is we're slowly diluting it out by flaming it, killing off all the bacterial cells that we got on. And then you start here and you streak through a little bit of the inoculum and then through that other streak, and you go back and forth. Like that. Flame it again, and then go streak through it again. And each time you come up and you touch the previous streaks until you get toward the end where now you're not going through any of your previous streaking, you're just streaking it on the plate by itself. Flame, and then do one last time here, okay? Blah, 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 blah. And what that does is, like I said, that dilutes it out so that you can now see this region here is where you put the inoculum initially. And now you can see that you started streaking it this way, and you started streaking it this way, and you started streaking it this way. And it dilutes it to the point where you now have these individual colonies. And they're different colors, and those colors are going to represent different bacterial species. Okay. Another technique for isolation is the pour plate technique, where you have a sample of inoculum and you take a little bit of it and you put it on the plate. And then you want to do serial dilutions where you dilute this inoculum from here. You take a little bit of this and you put it into a test tube that has no bacteria, and so that's going to dilute it out some. And then you pour a little bit of that onto the plate. And then you take this and you further dilute it by adding it to a new test tube with media in it. And so eventually you get it to where you're diluting it out to where you can pick these individual colonies. Because if it's too concentrated, you're going to have this situation here where it's really hard to pick just one colony without accidentally touching a neighboring colony next to it. Okay, ideally you want to have a plate like this where there are these easily discernible colonies. Like this one here, that would not be a good one to pick because this one here is touching this one here. Okay, and maybe it's the same bacterial species, but there could be mutations already that have arisen in those same species separately as a colony. So you want to get them by themselves. These would not be good colonies to pick from up here either, because see, they're all touching. You want to pick the ones that are not touching any other colonies so that you have a pure representation of that bacterial species. There's a spread technique where you, again, so this is your pipette, and you are going to use the pipette to inoculate the plate. So you pour the liquid that contains the bacterial cells onto your agar plate, and then you have what's called a hockey stick that you make out of a glass pasteur pipette. You just hold the glass pipette over the Bunsen burner and it starts to melt, and then it droops down, and it's a fun thing to make. And yet again, something that we'll be missing out because we don't have these face-to-face labs. But you would take that hockey puck and then you'd put it in the inoculum, and you'd either spread the hockey puck or you could turn the plate and just rotate it, and spreads that medium out onto the agar plate. And then, of course, like we said, we inspect our plates. You see, are these colonies, are there any colonies that I grew up that I can pick individually, right? Are there ones that are the right color that I'm looking for? And if so, you can pick those. Is there a bunch of contamination? So my technique wasn't good, and maybe I didn't flame my loop enough, or maybe I accidentally coughed in my plate, or I left the lid on my plate open for too long and the microbes in the air fell down onto it and contaminated it, right? That would be contamination right here, right? You don't want that. So you got to look at it and just see how the growing occurred, like you guys have done with this lab that we're going to talk about soon. Some of you got nothing, some of you got something. Some of it might be actually from your hands or coughing or phone. Some of it might just be contamination, that some microbes fell on the plate while you were inoculating it with what you were hoping to grow. Identifying a microbe, we talked about that a little bit last time. There are lots of different techniques. You can sequence the DNA, or you can sequence the RNA. So in the paper we talked about on Monday, they did a 16S ribosomal RNA sequencing to determine the different bacterial species in the gut microflora. You can do biochemical tests where it will, you inoculate it and it will turn a certain color based on the type of bacteria and what that bacteria excretes, or the byproducts of that bacteria. So remember again, when we talked about the paper on Monday, they were looking at downstream metabolites, and one was propionate, right? And they also looked at acetate and butyrate to compare the different species to see what they were producing. That would be another way to distinguish these bacteria. Do they make propionate? Do they make butyrate? Do they make acetate? And then immunological tests, so ELISAs, for instance, or what's called immunohistochemistry, where you will use antibodies to target proteins of interest and then dye those with a stain or have a fluorophore attached to it. When we are growing things in a lab, you can use different types of media to grow it. The plates that you had were called TSA plates, or tryptic soy agar plates. And those were used because they tend to be conducive to a lot of different bacterial species, okay? They're broad, they're not differential. They don't select certain types. They grow almost anything. It can be liquid, it can be a solid. You can use a broth that I talked about a little bit on Wednesday, how early microbiologists used to just use bone broth, right? And so we still use a similar media that's liquid to grow things up. It can be synthetic. So that means it's chemically defined. What do we mean by chemically defined? Well, if you have synthetic media, it's been made in a lab, and so they can tell you exactly what is in that media and how much of what is in that media, okay? Complex media is different. It's not absolutely discernible what's in there. Okay, so for instance, if you're going to grow something and you're going to use sheep's blood because there are hemorrhagic bacteria, right, that will break down the hemoglobin in their blood and on the plate, and that will show you that. You don't know every single component that's in that sheep's blood. Okay, we can infer what's the majority of stuff in the blood, but you don't know exactly everything in there and the concentrations and the amounts. Whereas synthetic, it's completely defined. Sometimes they'll use like they will grind up cow brain, okay, and use that as a nutritional source of media for growing microbes. And yeah, you don't know exactly what's in that brain, okay, that you use. So it's called complex. There's media that can tell us different things. There's enriched media, there's selective media. So certain things, if it's a selective media, that means it will only grow certain types of microbes. So if your microbe grows on it, you know something about that microbe. If your microbe doesn't grow on that, then you also know something about that microbe. Differential, it's kind of like a yes or no answer. Anaerobic versus aerobic, and so on and so forth. Okay, so the media that's used in the lab is full of variety, and all of those different varieties have benefits and have ways to inform us about the microbes that we're trying to, at the end of the 6 eyes, identify, right? Again, it can be liquid, it can be semi-solid, or it can be firm. And that's all you really need to know about that. So here we're looking at a culture tube of semi-solid, probably agar, just not as concentrated. So it's still it's gelatinous. It can be liquid, it can be completely solid. The agar is what we typically use the most for growing on solid things. That's what you guys picked up. For those of you who couldn't come to campus, you tried to make it yourselves. I don't know if that was successful for any of you, but I'm curious to see when we talk about the lab in just a little bit. It's solid at room temperature. And so the way you make agar is that you add the agar, which is a powder, to water. You boil it to kill off most things that might be in there, and to melt it. And then you let it really cool down to about 50 degrees. So you put it in a water bath at 50 degrees, and that's just high enough to prevent it from solidifying, but not so hot. You don't want to go straight from the boil and pour it into a plate, because then you'll get all this condensation that will then rise up on the top of the lid of the plate, and then that water will, fall back down into your culture. And so you want to cool off a little bit, but not so much that it solidifies, because at 42 degrees Celsius, that's when it turns to a solid, okay? The agar, it comes from seaweed, and the reason we use it is because most microbes, by and large, can't break it down, okay? As we talked about, microbes do all kinds of digestion benefits for us humans with fiber and other things, but the agar is one thing that they cannot break down. So they grow on top of it. They don't digest it and start migrating down. So you have a nice surface that has colonies on there for you. Nutrient broth, nutrient agar. Those are the two main media, okay? The broth is the liquid, the agar is the solid. And the components in there are usually beef extract of some degree and peptone, which feed the bacteria the nutrients they need to continue to reproduce and grow. This goes into, again, the synthetic versus the complex, or non-synthetic, general purpose versus enriched. Okay, so typically we'll use just general purpose media just like some agar. But if you want to use an enriched media, like I mentioned with the sheep's blood, to look at whether or not they break down your bacteria is hemorrhagic, then you could enrich that media with some blood, and that would tell you whether or not it broke it down. Okay. This is looking at that. So the Streptococcus pyogenes, it is a hemolytic bacteria. So so this is a plate, and the red is blood that you've added to that agar. Okay. And what you will see is you'll start seeing a white halo around the colonies. Similarly to the capsule stain that we talked about, only it's not because it has a capsule, it's because it's breaking down that hemoglobin. And so it's losing the red pigment as a result. Conversely, if you have this situation where you have colonies growing, but it's not breaking down the red media, then you know it's not hemolytic. Okay. And so that's a way of discerning what flavor of bacteria you have. Some things can be selective, some things can be differential, and sometimes you can have both, right? So depending on the color of your bacteria, that will be differential, right? Red, whitish, or greenish. That tells you something about that bacterial species. If it doesn't grow at all, that's selective. That tells you, well, this fourth one here, nothing even grew. So it does not get the nutrition it needs from this media to grow. And so that's a selective, right? Yes or no. Whereas differential is A or B. Does that make sense? Are there any questions on that, you guys? Anybody curious about, is anything unclear at this point? Okay. So let's try something here. Chrome agar contains several dyes and is used to diagnose urinary tract infections, okay? The patient's sample is inoculated, and based on the color of the colonies, you can identify the pathogen based on the color. So chrome agar is best described as what? Enriched, selective, differential, or complex? What do you guys think?
Differential.
Differential. Anybody else think something different? Are there any differential answers? Everyone thinks differential. Yeah. Well, differential it is, because it's the color, right? If it said that it was grown on a media that either grew it or didn't grow it, then it would be selective. Okay. Differential is going to be this color or that color based on the bacterium. Okay. And so, like, again, this is differential based on the color, also selective because some things don't grow at all. Okay. Last slide. There's other media that can tell us whether or not the bacteria does certain types of fermentation. So lots of bacteria are fermenters, and through the process of fermentation, you have the release of carbon dioxide. And so if you wanted to see if it was fermenting sugars, you would put it in a media that has sugar in it. And then if there's a bubble that's formed, you know that that bacterium is a fermenter because it's producing carbon dioxide. There are also things you can add, pH indicators. So if it's making acids, right? Because like we said, certain bacteria make propionic acid or propionate, or acetate, acetic acid. And so that will then change the color of the media if it has an indicator in there to detect a lower pH. So that's the worldwide web of media. And I think, yeah, that's the end of that lecture for chapter 3. So any questions on that or comments? Okay. I went through that, kind of fast. This is basic stuff, not the most interesting stuff. All of this is going to be much more relevant if we were in the lab doing these things, but it's important that you learn it because it is a major part of microbiology. Okay, well then, if there aren't any questions, let's move into the lab. Who has some results that they would like to share? You can show us your document, or you can, if you have your plates, you can hold up your plates. I'm curious what grew on people's cultures.
So I made my own dishes, and I actually had a question whether they were looking good. I have some pictures.
Awesome. Okay, so this is good. This will tell us whether or not the at-home, it's not really agar because you use gelatin, but if that works or not.
This was like one of the most ones
Okay.
I got them.
Yeah.
And then that one.
Okay. What was the difference between those in terms of the treatment?
One I think was the cell phone, and one was coughing.
Okay. And so presumably those were the ones that you didn't have a mask on and that you didn't that you swabbed before you used the alcohol wipe?
Mm-hmm.
Great. And how do they compare to the plates that you coughed on with a mask and the swab that you did after the alcohol treatment?
So the one with the alcohol treatment, there's barely some stuff. There is stuff, but not as much as the dirty one. And then with coughing with a mask, I can see only like a dot right there. That one's with the mask.
Okay. And so that's relevant, right? Because clearly not everyone thinks that masks are effective in what we're currently dealing with in this country and around the world. But it seems to me like that's pretty good evidence that a mask will trap microbes from coming out of your mouth. Anybody else? Great, perfect. So you did get a solid media and you got stuff growing on it. And the stuff that you didn't clean and the stuff that you weren't wearing a mask with had more growth, which is expected, right? Awesome. Who else? Anybody else had results that were similar or different?
I did washing machine.
Oh, interesting. Okay. And what did you see with the washing machine?
Well, let me see. I don't know if you can.
Oh, yeah. Okay.
So that's before alcohol and then after alcohol.
Nice, nice. And so we have some different colored colonies as well. It looks like there's some dark ones and some light ones and some yellowish ones. And then on the side that you didn't use alcohol, there's one little colony.
Yeah, it was pretty cool.
That's awesome. That's really great. So I know some of you guys did get creative, and that's awesome. So that's cool. We did cloth diapers for a couple of my kids, and so you know, if you don't have a diaper service, then you're washing those diapers in your washing machine, and then you wash your other clothes in the washing machine. And so luckily I never really felt like my clothes smelled bad, but clearly there are microbes in your washing machine, right? Even though it's counterintuitive, right? You think that'd be pretty clean because you're washing with soap all the time. Well, that gets into the point of sterilization and not sterilization, right? You can wash your hands, but you're not sterilizing your hands. You're washing off most of the bacteria, but not all. So interesting. Anybody else want to share their results?
Yeah, Doc, I think, oh, go ahead.
Sorry, no, go ahead.
So I was saying that I had a little cross-contamination with the cell phone, and one had dropped into the sink and caught some grime. And on the bottom, guinea pig poo. I apologize for the rookie mistake as far as the writing in the middle of the screen there, but
Okay.
guinea pig poo and pee on the bottom.
Yeah.
And so there seemed to be an explosion of colonies, and cool.
All right. So definitely, we talked about the sink being one of the dirtiest things in your household, and so clearly dropping your phone in your sink is probably worse than dropping it in the toilet, assuming that your phone can withstand the water and it's not damaged that way. But if you really are going to freak out about the stuff that's on that phone, going down the garbage disposal would be pretty gnarly. All right, Natalie, I think you were going to speak up potentially.
Yeah, sorry. So I have a little bit of condensation on my plate. I ordered some from Amazon. But this is the one for the index finger. I didn't really read the instructions correctly, so I ended up using a sterile swab and wiping it on my finger instead of just wiping my finger on the actual plate. But before and after, so this is over here is before.
Okay.
And this one's actually fuzzy and black. I don't know if you guys can see that. And
Yeah.
then obviously over here is after.
Interesting. So is the fuzzy one might be mold, actually.
Yeah.
Does it looks fuzzy? I mean, I don't open it or anything, but does it kind of look like furry in a way?
A little bit.
Yeah. So chances are that's going to be mold. And of course, mold is, or fungi, some type of fungi is everywhere. It's on our hands. And that could have been from the swab. It could have also been because mold spores are in the air all over, that it might have, when you were, when you took the lid off and you were inoculating the plate, that a mold spore fell down onto that. And you still have stuff growing, right? Even after you do the alcohol on your hands, you still have some culture there, right? You're not sterilizing, you're disinfecting, right? There is a difference, and we'll go into that, later on.
And then I didn't have anything. I wasn't sure if anyone else had the same issue, but I didn't have anything growing on my coughing one, like even without my mask. It just nothing grew.
Nothing grew at all. Okay.
No.
Anybody? So let's go there. Did anybody have cultures of one treatment, right? Whether it was coughing or the hand or the phone, but no cultures at all from another treatment? Like Natalie.
I did, and it was really surprising because I actually had a lot more growing with the mask, and I think it was because the bandana I used was actually dirty prior to that.
Okay.
And then the one without the mask, I have like one little dot on there that's barely growing at all.
Interesting. Okay. So your hypothesis is that maybe your bandana was just kind of dirty, and then when you were blowing air through that, it blew the bacteria that was on the bandana onto the plate.
Exactly.
Okay. What else? What else do we got?
I have one. This is my phone before and after. I don't know.
Yeah.
It's kind of a glare. So the before is on the top.
Yeah.
And it's really gnarly. There's a lot of different types.
I see.
Okay, there you can see it better. And then the after, there's nothing. So the alcohol actually did clean my phone really well.
Awesome. Yeah. So you have different colors. And so like we just talked about, those different colors and shapes are going to represent different bacterial species or even genera. Anybody get nothing? Who got nothing?
Well, I actually point out on one of my cultures.
Yeah.
So this one is my hands washing before and after.
Oh, okay. Before you say anything, who wants to guess? No, hold it back up there. What do you think? Which do you think is the right side, and which is the clean side? Which is the before clean side?
I'm gonna guess that the one with less is after she cleaned her hands.
So our left, your right Hava.
Yeah.
Is that
Yeah.
correct? Yeah.
It is correct. Well, I was actually going to add to this because when I washed my hands, I didn't dry my hands good enough with a paper towel.
Mhm.
And so this dot right here
Mhm.
is like a little bit of soap and water from when
Mm.
I didn't dry my hand completely well. But this dot right under, or this right underneath it
Ah.
was after I got a new paper towel and dried my hand more thoroughly. And
Mhm.
so this, it's interesting to see how non-washed hands and then washed hands without drying it properly and then drying my hand properly
Does,
had changed it.
does anybody know? Does that make sense? Do we know? So if we know what soap does, assuming it's not antibacterial soap, which you guys shouldn't be using, regular soap, you guys know how it cleans your hands? Anybody?
Yeah, it doesn't it break down the bilipid layer?
Yeah, well, so it forms what's called a micelle around that bilipid layer, okay? Because of the polarity of the water and the non-polarity of the cell, and you get these soap globules that then trap that bacterial cell. And then when you wash the soap off, you wash the bacteria off. So washing your hands isn't killing bacteria, again, unless it's antibacterial soap. If you're using just regular soap, you are just surrounding those bacterial cells and then washing them off the surface of your hands. Okay? And so the fact that you had that water droplet of the soapy water makes absolute sense. And that's really cool. That's a good demonstration of that. That what would have gone down the sink, that would have just taken all that bacteria off of your hand. Very cool. Anybody else want to share what they have or don't?
I have a question.
Yeah.
Only one of my plates had a lot of condensation,
Okay.
and I wasn't sure why that happened.
Well, so these plates, the auger is made with water, right? I mentioned in lecture that you add this powdered auger to water and you boil it. And then what happens is eventually that water is going to evaporate out of the auger, which is why we usually turn the plates upside down. And that way, if the condensation, were your plates lid up or were they lid down? Or do you recall? Oh, you're muted.
Yeah, they were lit up.
Yeah. So lid up means that the water that evaporates out of that auger then evaporates and condenses on the lid. Okay. And so the way we get around that is we turn the plate upside down. So that way, if it evaporates, it just evaporates into the auger that's already there. And that's why a lot of your plates, I think, kind of dried out a little bit, unfortunately, too. Did anybody have plates that just dried out beyond recognition? Okay, good. I know that was a problem, it seemed like, for some students. They had a very thin layer of auger that they could barely even see. Okay. Does anybody want to share a horror story? I mean, not a horror story, but like a mine didn't work at all kind of a thing. There's no penalty for this, right? There's nothing wrong with it not working at all. It didn't mean that you screwed up per se.
I kind of feel like mine didn't work at all.
Okay.
I mean, there's some growth, but it's not really on the base. It's like, it almost makes me feel like it was cross-contaminated or something, because it's like just on the edge only.
Okay. Yeah, and presumably you didn't touch the edge.
No, I mean, not to my knowledge. I tried to be really careful, so I don't know what I did, but.
Yeah, that happened to mine too. It just like all around the edges, and this is the only one that I got anything on, and I don't know if it was from maybe like stuff seeping in the cracks or what, but yeah, it's weird.
Typically the plates should form, I mean, it's not hermetically sealed or anything, but it should close off and prevent bacteria from coming in, especially if you then seal it with the parafilm. Yeah, that's interesting. I have not really noticed a whole lot of edge effect of contamination before. So I don't know what that is, but that's
Mine was the same too, where like my other ones didn't have any growth, and then like hers, and then just the one had it all around the edge.
well, maybe, who knows? Maybe the lab tech did something that contaminated those, and you guys were the lucky ones to get them. I don't know. Hard to say. Good though. Anybody else have anything they want to share?
Yeah, I didn't have any growth whatsoever in any of mine,
Okay.
which was disappointing.
Absolutely. I feel bad.
Me neither.
Yeah, I know. I know some people didn't grow anything. Somebody's media didn't solidify, or it did, and then it turned into liquid. You know, and so that's just it's so anticlimactic to go through the motions and then see nothing. Which, you know, by all manner of expectations, we would think that there would be something, since microbes are everywhere, right? It's like, how did it not work? So my encouragement to you guys who have nothing, if you want to try to inoculate it again, if you still have enough of a auger pad on there that, you know, it could just be the conditions for whatever reason. It was not warm enough in your house or something. You could try to just inoculate it again and just say, okay, I'm going to get something that I know has bacteria on it, and I'm going to stick it on there. And by golly, if nothing grows, then there's something wrong with this media, right? That would be my recommendation. The hard part.
Oh, sorry.
No, go ahead, Christine.
I was really interested to find out if the people who made their own, for the most part, did you guys get growth or not?
Yeah, I did.
Yeah. I know one person that did the at-home media. It turned to liquid. So I think who else? Anybody else do the at-home, made their own, and got something to grow?
Mine completely liquidated, and I tried mine 3 different times.
Oh.
So it was kind of a fail.
Okay. Thanks, Ashlyn. Okay, so we see these things, we see differences, and this is why we have a lab, right? Because you all presumably are living in different environments, right? And they're all have their nuances, right? And we can't control for this. And so in the lab, you'd all be doing it in a relatively, you know, same consistency, and we'd be putting it in an incubator and growing it at 37 degrees, which would speed up the rate of growth. And we could then have fairly similar results, right? Fairly reproducible. But because there are all these variabilities from one home to the next, and from one, you know, making it one person's hands to another person's hands, then we see this, okay? So for those of you who got nothing, I still want you to put your images. You got frustrated and decided to microwave it. What did that do, Brandon? What did the microwave do? Just melted the auger?
Didn't do anything. I wasn't trying to microwave it, but since I didn't really see any growth, I decided to just kind of put in there for like 5 seconds most. And then it kind of didn't do anything. It kind of just like it turned yellow from the sides a little, you know, like around, just like that. Yeah, that's all I did, but it didn't really melt.
Well, my recommendation is that for those of you who did get stuff, if you want to treat your plate with a little bit of bleach and then throw it away, you can do so. If you want to, if it's sealed with that parafilm, then you can just pitch it. It should be fine. If you didn't get anything, but your plates are still in decent condition, you can try, just for your own curiosity, inoculating them again. And for those of you who didn't get results, still put your pictures into the Word doc and explain it, and maybe hypothesize why you think it didn't work. Or if you have no idea why it didn't work, just state that and say, maybe it's like, with Hava's result, she should mention that water droplet from the soap, and think of this as you guys conducting an experiment, not just doing a lab. Labs are not experiments mostly. Labs are just demonstrations. They are based on activities that are tried and true and are very successful. They don't really give you an idea of what science is like in the real world, right? When I got out of college and then I started researching in a lab, I was very, very shocked that my things didn't work, right? And so this clearly had some success and some failure, and treat it like that. Treat it like an experiment and write out some results and what your conclusions are if you want to. If you want to just answer the questions, then by all means do so. Hopefully you can answer the questions even if you didn't grow anything, because it would be based on what we've talked about, the ones who did have successful growth. Okay. And if you have any questions on that, feel free to shoot me an email, or you can ask right now. Anybody have any questions on the lab itself? Because it's due the end of the day today.
I've got one for you, Doc.
Yeah.
Step or number 17, use the morphological key to describe the colony and colony morphology.
Mm-hmm.
Where's that hidden key at?
Let me see. Did that not get included? Where was that, Ryan? What part?
I'm on page 6,
okay.
Number 17.
Okay. Yeah, just skip that.
Okay. Thank you.
Yeah.
Sorry, go ahead.
For table 2, it wanted us to put in class averages.
No, average amount of bacteria before and after hand washing. So you'll use that key, amount of bacteria 0 to 4. So let me share my screen. Okay. So what you'll want to do is you'll want to look at this section here. Okay. So 0 being no bacteria, 1 being little, 2 being some, 3 being more, 4 being substantial. Okay. So for table 2, I would just don't do the whole class, just do your own, because we can't really all get, it's too much to just to do that. So I would just say left index finger before washing. If you got nothing, it would be 0, right? And then this is highly subjective, right? What you think is a 1 may not be somebody else's 1, but that's not the point. The point is just to get you guys thinking about quantification.
And then I was looking at table 2 as well. It says, after washing with water and then after washing with soap. Do we just put soap and water?
Yeah, because it was the same,
Okay.
it was basically the same treatment that we combined into one. Yeah.
Okay.
Yep. Any other questions about that? So in the conclusions, this is what I was kind of saying before. This is where you would kind of speculate, right? This is your opportunity to say what went wrong or what went right. Why did it go wrong? Ashlyn, you can say, I tried making it 3 times. And so I had a statistically significant failure at making the auger, right? Because as they say, if at first you don't succeed, do it 2 or 3 more times so that it's statistically significant. Bad joke. That's a bad nerd bioscientist joke. But anyway, okay. Any other questions on that? On the lab?
Yeah, I have a quick question. So technically, I don't know, like if the growth around the edge is really considered growth since it wasn't necessarily, you know, I don't know what it was.
I wouldn't use that for table 2 where you need to quantify it, but in your results, in your conclusions, just mention that. Say, I had some growth around the edges of the plate, and then speculate as to why you think that is. You know,
okay.
Contamination. Where do you think the contamination came from? It came from you. It came from before you had it. You know, that's up to you to, you're the scientist here. So your conclusions.
I'm curious, does the growth actually slow down after a while, or is it just that it doesn't have its nutrients as much anymore because it's eating away at it, at the agar?
Yeah, Ray, we will talk about growth, bacterial growth, and there's a log phase, there's an exponential phase, there's a stationary phase. So after a while, your bacteria will just stop growing. It will slow down as the nutrients kind of get exhausted from that area. And absolutely. If you left those plates out, they probably wouldn't grow a whole lot more. Unless you reintroduced bacteria, new bacteria onto it.
Are we just emailing this document to you, or?
No, I think, did I not create an assignment? There should be.
You might have. I might have just not seen it yet.
Okay, let me make sure that I did that. But yes, you just submit that document. Yes, there's a growth in morphology lab assignment, and it's published. So just submit it to that.
Okay, thank you.
Yep. All right, anything else, guys?
I have a question about my plate. So it kind of came out a little foggy, but you can still see all the bacteria. So that's my brother's little toy. And I don't know if you can see that. I tried to do it with a flashlight. Maybe you guys can see it better.
Oh, there we go. Yeah.
Yeah, and that's my phone. Hi.
Okay.
Yeah, so it looks like my brother's toy was pretty bad compared to my phone.
Doesn't surprise me.
Yeah, he showers with them and all that stuff.
Yeah, I was just talking with my mother-in-law before class because she's here helping me out with my kinder pod while I teach. You know, and how the daycare that my 2-year-old goes to, like, they don't really rotate the toys out. They don't have toys one day and then take them out and put some new ones in. It's just there, allowing the kids to cough on and lick and, you know, do whatever stuff they do to these toys. And then that becomes a vector for the next kid that walks up to it. And then they become vectors when they come home and give it to all of us. So yeah, children's toys, they go everywhere, literally, right? And so I think that was a cool experiment, you know, because phones are considered very, very unhygienic because they're everywhere. They're on your face. Then you set it down on a desk somewhere, and you know, then they jam them in your pocket, and there's all kinds of gross stuff that goes in your pockets and that doesn't get washed out of your pockets. And so phones are pretty bad. And that was before you swabbed with the alcohol, right? That was the dirty phone.
Yeah, so it was a dirty phone and the dirty toy.
Pretty neat, huh? I'm glad you were sharing. I was hoping that you'd share and let us see.
Yeah, my mom just disinfected everything after I showed her, so.
Yeah. So let's see. How do you put the photos we took in the doc? So you should be able to just in Word just go to Insert. So you want to use your mouse and just click on the area where it says to insert, and so that you have your little bar blinking there, and then there should be an insert option. So just go to Insert and then find the file and put it in there that way. I think you can also copy it and paste it as well if you want, but yeah, so just integration, exactly.
I had a quick question. How would condensation on the top of the plate, say if like some water, because I had condensation in mine, but I haven't had any growth yet. If water got onto the auger, how would that affect the growth?
Yeah, that's a great question, because that's why we do turn the plates upside down and incubate the incubator upside, because if you imagine at room temperature, you're getting evaporation. When you put it at 37 degrees, you're going to get even more evaporation. And so what would happen is if that plate on that top of the lid wasn't completely sterile because you opened it, you know, to inoculate and something got on it, then the bacteria or the water that condenses on top would be inoculated with contamination on the top of the lid, and then eventually that droplet would get so heavy that it would drop back down onto the auger, and then you would get, you know, inoculation that wasn't from your specimen, your collection. Let's say your lid was sterile. What the water could do is it could just drop on your bacteria, and then it kind of dilutes it out, and then you don't have those nice colonies for picking. Right? And remember, when we're growing up these plates, the whole point to do it is to isolate and then take it further. So you'd want to be able to pick a colony. Well, if it's got a big old water droplet on it, they all start to bleed together, so to speak. Does that answer your question?
Yeah, it does. Thank you.
Yeah. Any other questions or comments or thoughts on this?
Yeah, this might seem like an elementary question. The next step then, if I want to see this closer, besides doing the magnifying glass, and now we're talking microscope, can you lead me through the steps? We pop the lid off. Can you extract it from the auger and actually get a decent sample to smear on a slide?
Yeah, so there are several things you could do.
Thank you.
If you wanted to just stain it and just see what it is, you could take a loop and then you could touch it to that colony, and then you put a little drop. Usually, you put a little drop of water on your microscope slide, and you take your loop and you mix it around into that drop of water, which kind of dilutes it and spreads out the bacteria. And then you hold it at an angle and you let the water kind of roll down the slide to spread the bacterial cells out. And you can look at it just like that. You could put some methylene blue on it just to look at the bacterial cells to see if they're, say, rods or if they're circular. You could even do these differential stains. You could do a gram-positive stain, or a gram stain, see if it's gram-positive or negative, an acid-fast stain. And then you look at that under the microscope. Okay. Another option is like, some of your plates, you have these big, huge globs, right? I would want to probably further isolate that out. So I would take, right. Yeah, exactly. So what I would do with that is I would take a little bit of that and streak it on a plate. Okay. So going back to that slide deck, let's see, going back to the slide deck, hold on a minute. Where is it? Where we were streaking the plates. That is what I would do to further separate it out. This one right here. Okay. So you could take a big loop of that and then put it on the edge of the plate, and then get your swab or your inoculating loop and then just streak it out and see if you can get more refined colonies. Because it's possible that that's all just one type of bacterium. It's also possible that it's just a bunch of different bacteria that are growing together and happen to look the same outwardly, happen to have the same macroscopic morphology, color, shape, and stuff. So that's what I would do next with that plate in particular, Ryan. I would try and isolate it out further. Any other questions?
Sorry, I noticed the agar actually provides luminescence to different strains too. When you look at it under the light, I'll get like yellow, blue, and red depending on the colony.
Really?
Yep.
Depending on the colony, you'll get different colors. Yes, absolutely. You mean like the way the light hits it, or just the colony is always one color?
Here, I'll show you real quick.
Oh, yeah, let's see it.
I don't know if you can see that, but I do get two different types of color, especially on some other ones. I'll have, I think this one was like a yellow or a blue.
Mm-hmm. So kind of some iridescence. You know, Ray,
Yeah, exactly.
It's highly possible that you just have a gamish, if you will, of different bacterial species in that section. And so as you kind of hold it out, you see some of the red bacterial species, and then you see some of the yellow ones, and they're all kind of there because they haven't isolated out, because it's because of the concentration.
Well, it's the concentration that I'm talking about, though, because like with larger colonies, it is just one color. With the smaller ones and with the thinner layer of bacteria, you can see through it, and certain blobs give off different colors. Like it's like per petri dish, I have different colors. Like I have a blue one, I have a red one, and I think a yellow one somewhere. But yeah, it's just really weird that I noticed that under the lighting.
Interesting. Yeah. So that's great, because that shows you a little bit about the diversity and the variety that you were able to capture on that plate that may not always be there, depending on what was most prevalent on the sample that you provided, that you collected from, or what just grows at room temperature even. You know, a lot of these things that are on our body, like on your finger, it's used to being obviously the outside of your finger is not as warm as internally, but it's still a little bit warmer than room temperature. And so you may not harvest the things that thrive in a warmer temperature than room temperature. Okay. Anything else, guys? We got a couple minutes left. We didn't get into the research proposal. So what I'll do is I'll just go ahead with what I was planning and just make a recording of myself going through the template, and then you can watch it. We'll need to have topics by next Friday. So you got a week to percolate, look at the news, look at social media. If you're struggling for an idea, again, it doesn't have to be clearly defined right now. Maybe it's something as simple as I want to work with bacteria, and I want to work with MRSA, right? Because it's prevalent, and I want to go into nursing, and MRSA is a big issue at hospitals. Or I want to look at nosocomial infections, you know, hospital-derived infections and things like that. Or, you know, maybe you want to work on viruses. So, you know, of those 6 microorganisms, pick the one that is calling you, whether it's fungi or protozoans, right? Or viruses or bacteria, or algae, right? Maybe you're interested in trying to improve the global climate change, and you want to use algae to make alternative energy, and you know, you want to make it more efficient or something like that. So prions are proteins, misfolded proteins that will then enter and cause your native proteins to misfold as well, leading to plaques in areas that are just hollow.
Are they considered like a virus?
No, they're not a virus. They're a particle. You know, they're an infectious particle. Like a virus, they're not alive, but unlike a virus, they don't have genetic material. They're just a misfolded protein. You're welcome to do something on prions if you want, because it's not a microbe, but it's an infectious agent. So pick something and then try to, narrow it down into, pick your microbe, and then try and focus on what you want to do with that microbe. And then hopefully, we'll come to something. Also, I posted the reading for Monday's Journal Club. It's an excerpt from The Immortal Life of Henrietta Lacks. I've kind of edited it so that, I tried to keep it as short as possible. So hopefully it's consistent and fluid, but some of the sentences I've removed and stuff like that. So bear that in mind. But when you read that, think of the, it's going to be different from this week's Journal Club where we're critiquing a science paper. For this one, you're reading it, and I want you guys to think about the social implications of what went on. And, this happened in the early 50s. How would that be different today? And, while there were a lot of good things that came out of getting these cells, what were some of the things that were not right about this whole process? And and what I'm trying to do is I'm trying to focus on this because it's something that happened to a woman, and an African American woman, in a time where we were very, very racially divided, similarly to what we're experiencing now. Worse in some ways, I guess you would argue, for sure, but still it's an issue. And hopefully we don't see this in the medical or research realm anymore, but it happened not long ago. And so with that in mind, as you're reading this, kind of think about that. That's what I'm looking for. I want to discuss it from that perspective. And we'll talk about it Monday. But any other last minute questions or comments? Homework's due Monday. Some of you have already turned it in. Anything else? Okay. All right. Well, in that case, thank you guys for showing up. It's been nice chatting and seeing you, and I hope you have a great, safe weekend. I'll see you Monday at 10 o'clock. We'll have lecture, and then we'll talk about our reading for that day. Okay. Any feedback you guys want to give me on the quiz is welcome. Otherwise, we'll just plan on it being like that. I think it was great. Everyone was able to access it and stuff, so it worked out. I've never done that online before, so hooray. And yeah, I'll hang out for a couple minutes if you guys have something you want to bring up. Otherwise, I'll see you next time.